This is part 3 of the chapter 4 lecture notes. And we left off, we were talking about eukaryotic cells and the differences between animal and plant cells. So this is a typical animal cell. It doesn't mean that all animal cells look like this. There are variations. They're not all rounded. Some of them have long extensions like nerve cells, and some are just very elongated and like more like a cylinder than a round cell. But anyway, this is a typical animal cell. And so you can see the cell parts, and I wanted to point out the cell parts that animal cells have that plant cells don't. So lysosomes are the organelles that contain digestive enzymes, and they are able to digest food as well as there are lysosomes that can help digest old, worn out red blood cells, or they can break down bacteria or other pathogens that enter the body. A lysosome can even help a cell to commit basically cell suicide, or it's called apoptosis, just by bursting and releasing its enzymes, and then the cell will just digest itself. But plants do not have lysosomes. They make their own food, so they don't have to break down larger food molecules into smaller ones. They make their own simple sugars. So lysosomes are not really needed. So a plant cell does not have lysosomes, and a plant cell also, I'm looking, does not have centrosomes. And the centrosome is just the name for the two centrioles. Each centriole kind of looks like a stack of logs. So it's two of those they're kind of at a perpendicular angle to each other. So a centrosome is simply two centrioles, and I will write that so you have the spelling. So two of those centrioles make a centrosome, and plant cells don't have that. And the purpose of a centrosome or centrioles is to assist during cell division in lining up the chromosomes and then splitting them apart so that each new cell has a copy of the chromosomes from the old cell. So plants are still able to do that. They just don't use centrosomes to do it. Let's see, we can see ribosomes that we discussed. Ribosomes make proteins. We can see the nucleus with the rough ER attached. Rough endoplasmic reticulum contains ribosomes attached. And so rough endoplasmic reticulum is a network of channels that sort of functions like a little factory. The ribosomes produce proteins, and then the endoplasmic reticulum will modify those proteins. Remember, proteins have to fold and form three-dimensional, very complex shapes. So the rough endoplasmic reticulum is like a little factory that helps fold the protein into the correct shape. And then we also have smooth endoplasmic reticulum, which produces carbohydrates and lipids kind of in the same way, like a little factory. And then no matter if it's a protein or a carbohydrate or a lipid, it ends up in the Golgi apparatus, which modifies and packages the product, whether it's a protein or lipid or a carbohydrate. It packages it and determines if it's going to remain within the cell or if it's going to be sent outside the cell. Vacuole is a larger storage molecule. A vesicle is a smaller storage molecule that is produced when the Golgi pinches off the very end part, like this part right here. You can see it kind of bulges. Well, it can pinch off, and then it would surround the product. And so the Golgi vesicle is what the product is packaged inside of, and and it's how the product is transported. Mitochondria produces ATP energy. The plasma membrane is a phospholipid bilayer that encloses the cytoplasm. The cytoplasm includes the cytosol, which is the fluid, and it also includes everything else. It includes all the organelles as well as the cytosol. Chromatin is the DNA located inside of the nucleus, the DNA combined with histone proteins. The nucleus, of course, the nucleolus is the structure inside the nucleus that makes the ribosomes that makes the ribosomes out of ribosomal RNA and proteins. And let's see, peroxisomes help to break down, they're kind of like lysosomes, but just for fatty acids, just they break down fats. And then you see these filaments or fibers that form the cytoskeleton. The intermediate filaments, the microtubules, and the microfilaments, these are just fibers that form the cytoskeleton. They play different roles. They can help in movement. They can just help give the cell sort of a shape, and they can help to organize the organelles so that they're not just floating around everywhere. But we'll talk about the cytoskeleton in more detail later. So we know what animal cells have that plant cells don't have. Animal cells have lysosomes and animal cells have centrosomes. Plant cells have some structures that animal cells don't have. Plant cells have a cell wall outside of the plasma membrane. Plant cells have a central vacuole. Now, animal cells have a vacuole, but it's smaller. Plant cells have a large central vacuole that's filled with a watery material. It's called cell sap, and it helps to maintain pressure against the cell walls, pressure called Turgor pressure. The cytoskeleton is still there in a plant cell. Plant cells have organelles called chloroplast. Animal cells do not have these. Chloroplast carry out photosynthesis, which is using sunlight to generate glucose, generate food. Now, just because a plant cell has chloroplast and makes its own food, it still needs mitochondria to convert that food, those simple sugars, into ATP energy. So plant cells contain cell walls, central vacuoles, and chloroplast. And animal cells contain lysosomes and centrosomes. Okay, so here's a large view of a centrosome. Each centrosome consists of two centrioles, and they lie perpendicular to each other or at right angles to each other. The organization of a centriole is these structures here. This whole cylinder is a microtubule. So there are groups of 3 microtubules. There are 9 groups of 3, so we call them 9 triplets. And they're bundled together. And the other proteins, which you see in green, are non-tubulin proteins that hold the microtubule triplets together. So in animal cells, these centrioles make up the centrosome, and they assist in cell division. In, let's see, the plant cell wall, which is outside of the plasma membrane, gives it extra protection. It is composed primarily of cellulose, unlike the cell wall of bacteria, which is composed of peptidoglycan. Chloroplasts are found in plant cells only, not in animal cells. And the fluid filled inside of the chloroplast is called the stroma. And these stacks, they look like little green discs. Each disc is a thylakoid, and then each stack of thylakoids, almost looks like a stack of coins, is called a granum. So one disc is a thylakoid, a stack is granum, the chloroplast carries out it has two types of reactions: the light reactions and the Calvin cycle reactions. So the reactions where light is used, pigments gather light, that is carried out in the thylakoids, whereas the reactions that use the energy from light to produce glucose or some form of simple sugar, those happen in the stroma. So the thylakoids are where the light reactions occur, where the pigments, the green pigments called chlorophyll, capture sunlight. Sorry, I didn't mean to make that line through reactions, but and the stroma is where the reactions, Calvin cycle reactions, occur, and those are the reactions which actually convert carbon dioxide into glucose or some simple sugar. Plant cells have a central vacuole. It is large, and this is different from animal cells. And the central vacuole helps regulate water. When a plant cell is placed into a hypertonic solution, that means the solution on the outside has more solute in it, it's more concentrated. I'm not going to write that whole word out, but a hypertonic solution is that when the solution outside that plant cell is more concentrated. So, for example, if we tried to feed our plant salt water instead of fresh water, then it would cause the central vacuole to lose water, and it would cause the plasma membrane to shrink away from the cell wall. See how it's all shrunk away, and basically it would kill the plant. If the plant is placed into a solution of that's isotonic to the plant, um, so isotonic to the plant would have the fluid inside the plant, or the solution inside the plant would have the same concentration as the solution outside, then there's not going to be any change in the size of the central vacuole. It's just going to be normal size. And you can see the plasma membrane, but it's more close to the cell wall. Here's the plasma membrane, and it's not right up against the cell wall, but it's close to it. Then if you put the plant in a hypotonic solution, like we do when we water plants, the water has a lower concentration than the fluid inside the plant cells. So pure water is hypotonic, and it's going to cause water to enter that central vacuole and make that plant cell swell up and cause what is known as turgor pressure. And that cell wall will keep the plant cell from bursting. And the way the plant would look is in a hypotonic solution. When we water the plant, it'll kind of stand up and be more rigid and look healthy. So we'll talk about that a little bit more in the next chapter. Endosymbiosis is a theory, a hypothesis, that says that mitochondria and chloroplast at one time, early in history, early on in evolution, when there were only unicellular organisms on the earth, that mitochondria and chloroplasts were actually prokaryotes. So they were actually some type of unicellular organism like a bacterium. And then over time, a larger cell, a larger plasma membrane engulfed the mitochondria and the chloroplast. And if it just engulfed mitochondria, it became more of an animal-like cell. And if it engulfed mitochondria and chloroplast, it became more of a plant-like cell. If you want to look into that further, Lynn Margulies, someone that you can Google, but and there's nothing else in this chapter about it, so it's just saying because the mitochondria and the chloroplast have their own DNA and they have their own ribosomes, and they're very, very similar to prokaryotic cells, that maybe they originated as those cells.